Agora, eu também estou a fazer uma gravação na casa de banho, por isso não chama.
Hello, hello everyone. Good morning. We just waiting one more minute to see if there's anyone else coming, and we'll start. Okay, so thank you all for coming to this second Congen talk. Today, Vasco Barreto is the coordinator of the Congent Working Group on Molecular Biology, but also a PhD in immunology from the Institut Pasteur in Paris. Then he went to the Rockefeller University in New York, first as a postdoc, then as a research associate, working with Michel Nussweins on the molecular mechanism of antibody formation. He returned in Portugal in 2008 to start his own group at IGC, and then he moved then to CEDOC at Nova Medical School, where he's the group leader and DNA breaks lab, but also assistant professor at the Faculdade de Medicina, Faculdade de Ciências Médicas. His group has developed a lot of work in CRISPR and cell lines, and it did something a bit different than the usual research groups do, which was trying to merge the need of funding with the willing to develop technology. And he's going to talk about this adventure that started a bit longer than a year ago, where to create a service on CRISPR for research institutions from scratch. So Vasco, please go ahead.
Thank you, João. Good afternoon, everyone. As João was telling you, I'm going to tell you about the first year of activities of our CRISPR CEDOC service. Essentially, what we do is the editing of conventional cell lines, induced pluripotent cell lines, and also primary cells, all mammalian cells, with the tools of the CRISPR technology. And I decided to structure this presentation this way. Can you see the slide? Is it okay? João?
Yes, yes.
Okay, good. So first, I'll just overview very quickly what CRISPR is. I'm sure most of you are aware of this technology and how it was developed in the first place. Then I'll tell you about the CRISPR at CEDOC, why we started it, when, and what we have been doing. I'll also show you some numbers which are important in the context of congenital. And then we'll go over a few projects in which we have been involved, and also a number of collaborations. And finally, I'd like to conclude with a few concerns and a few ideas so that the service can survive and thrive. What is CRISPR? So unless you've been living under a rock since 2013, this is probably not news to you. It's basically a revolution in gene editing that was noticed by the scientific community and also by the general press, as you can see by the number of covers that CRISPR has received. And these are the so-called heroes of CRISPR. So these are the people that developed the technique that we use today. And as you know, two of them got the Nobel Prize recently. CRISPR is basically an acronym for this very long name. And this very long name basically describes not a technique, but a type of immunity that bacteria evolved over millions of years that in a way mimics the adaptive immunity of vertebrates in its two key features, namely specificity and memory. And the way bacteria achieve this type of immunity is by keeping pieces of sequence information from the phages that try to invade them. They store those pieces in the genome, and then upon new infection by the same phage, the phage DNA is going to be attacked by a system that has endonucleotide activity that will destroy the entry DNA species. And this is done by the CRISPR system, which is able to capture the sequence of the phage, put it in the genome of the bacterial in a cassette array, and then, sorry, this wasn't supposed to happen. Let me just go back. In this array, and then once the DNA is encoded here, CRISPR can produce these endonucleases, which are composed of a protein that is able to cleave DNA, and a guide RNA that is going to give target. It's going to target the protein to a given position in the phage DNA. It's a very complex system, and there are many variants, as you can see in the right image. Of course, we won't be talking about these variants. We are mostly concerned with the biotechnological applicability of CRISPR. And in that context, it should be emphasized that in spite of all the hype around CRISPR, we should honor a number of scientists that long before CRISPR appeared were already able to do very precise gene editing. As you know, in 2007, the Nobel Prize went to these three gentlemen here for the discovery and the invention, actually, of a gene targeting protocol to target the genes of the murine genome. And the technique was essentially, and is still used, and it's based on the principle of homology recombination. So if you flank whatever piece of DNA you want to put inside a genome with sequences that are homologous to sequences in the genome, then you can promote a recombination event that will flip the original sequence in the genome with a sequence in the insert that you want to put in. If you add a selection marker, then you can enrich. This keeps happening, I'm not sure why. Then you can enrich for the cells that integrated the piece in the right place. Of course, then you need to genotype and so forth. So it's a complicated process, but it works, and you can put any piece of DNA eventually anywhere. Then we had over the last 2 decades, 3 major developments in this technique. Why? Because the frequency of integration is typically very, very low. And one way to increase this frequency of integration is to introduce a double strand break near this region. If you introduce a double strand break, then you are going to promote the DNA repair mechanisms. And by promoting it, you can increase enormously, in orders of magnitude, the chances of integrating the piece of material that you want to put in. And this was initially done in 2003 using zinc finger nucleases, which have basically this structure. It's basically a fusion protein made of part of an endonuclease that will cleave the DNA, and an array of these zinc finger motifs, each able to recognize a very small stretch of sequence. And by combining these different modules, you can make the endonuclease able to recognize a sufficiently large sequence of DNA so that it will be specific and it will cleave only or mostly in the desired region. Then in 2011, again, based on the same idea of combining these modules of proteins that are able to recognize very short pieces of DNA, the TALEN system was invented. And of course, in 2012, 2013, we had this revolution that was CRISPR. The main difference between these 2 first systems and CRISPR is that CRISPR is much cheaper, it's much easier to do. And the reason is that the specificity of the CRISPR system is based on the interaction between the target genome and the guide RNA, and it's very easy to order or to synthesize a piece of RNA, whereas it's very difficult to assemble these modules that are the basis for the zinc finger and the TALEN technology. That's the main difference. In terms of performance, the techniques are actually equivalent. So the success of CRISPR is essentially a consequence of its simplicity and low price. And of course, as you probably know, this discovery started in 1987 in Japan. Then Francisco Morita, a neighbor of ours, was the first one to recognize that this CRISPR system was in fact a type of immunity. And then there were a number of findings that would eventually give rise to the Eureka in 2012 and to the application of this technology to edit the genome of cells. Namely the first publication of Charpentier, also the collaboration between Charpentier and Jennifer Doudna, and then of course almost at the same time the papers of Zhang and Church on the possibility of editing mammalian cells, which is what we do at CEDOC. We created this service last year following a survey that I sent to many different groups all around the world, inquiring about the possibility of having a CRISPR service at very affordable prices. And the feedback was quite encouraging. So we decided to go forward with this. And it has been a sort of adventure because our background is not really in interacting with clients. Normally we try to get money from FCT and other agencies, and this is a completely new game. We are essentially functioning as a company and interacting with clients. Our strategy was to propose very low prices, and you can see the prices there. So a knockout in a regular cell line is about 2,500 euros, and a knock-in is about 3,500 euros. If you're working with kind of tricky cell lines, such as induced pluripotent cell lines, then you should add 500 euros. If you want to use a fancy Cas9 to edit the genome, introducing fewer off-targets than the regular Cas9, then you should also add 500 euros. But the bottom line is that the prices are still quite low, even when you go for the premium option. Let's put it that way. And initially, so starting in October, we got several orders from all around the world, as you will see shortly. The team was small at the time. Paulina, Afonso, and João Proença were taking care of the work. And then, as you know, COVID-19 came. We stopped having orders. Also, the productivity of the service went down tremendously, and it was only around late September, early October of this year that we were able to restart the work. And by then, we had so many orders, and we were so delayed that I had to hire a lot of people, as you can see, so that the service now is being taken care by this team of 8 people, including 3 postdocs and sorry, actually 4 postdocs, a PI, and 2 technicians and a student. And in terms of numbers, we have so far 33 clients. We have a few more in the recent weeks, but that's not really important. Most of our clients are from Europe. Portugal is the European country from which we have more clients, as expected. We also have a few clients from the United States and 1 client from Asia, from Saudi Arabia. In terms of revenue, so far we received 71K, and we expect to receive 113K in the coming month. So this is the money that is already contractualized, let's put it that way, because initially the client only pays 25%, and the remaining 75% will be paid upon completion of the work. So if we manage to finish all these projects, we should receive this 113K within the next 6 months or so. With respect to the type of projects that we've been developing. It's mainly knock-ins and knockouts, and one project in which we are doing a library screen based on the CRISPR technology, which has been very, very exciting to develop. With respect to the types of cell lines that we work with, it's mainly regular cell lines, your 293T, your EULA cell, but also induced pluripotent stem cells. And in a few projects, we have also been trying to edit primary cells, namely fibroblasts. The typical knock-in, knockout project starts with the design of the guide RNAs and the oligos for genotyping. For genotyping, we use the T7 endonuclease assay, regular PCR, and also the ARMS PCR, which allows us to distinguish between the wild type sequence and the edited sequence, even when the editing is very subtle, meaning one base or two bases. And first we designed the guides, which are represented here by these green sequences. We also design the genotyping oligos. Typically, the way we do it is by having external primers that should always give you an amplicon. So this is our positive control for the amplification. And then one primer for the edited sequence and one primer for the wild type sequence that will combine with the external primers and will give us the wild type band and, if present, the edited band. Then what we do is to optimize the transfection conditions, because we receive so many different cell lines. We have to optimize each time the transfection protocol, which is typically done by electroporation. Then we genotype the transfected cells either in bulk or as clones, and for that we use the T7 endonuclease assay, which, as you probably know, recognizes some type of mispairing between the two strands that will be a consequence of the break and the repairment of the break after the action of Cas9. And as you can see here in the bottom, if there was an editing event in the presence of T7, we get this signature band, which is not present when we don't have T7, and it's also not present when we have no guide in the transfection mix. So it's a very simple readout. It only tells you that the cells were targeted. It doesn't tell you that in the case of the knockout and in the case of the knock-in, that the right edit is there. It's an important confirmation to have. We can also do this, as I was telling you, this ARMS race, ARMS PCR, in which we can identify different types of bands. The top band would be the control band that should always be there as a positive control. And then this band or this lower band representing the non-edited and the edited sequences. This we can do in bulk and also at the level of clones. And eventually at the level of clones, we need to confirm by genotyping that the edit is there. In this case, we are looking at a single base pair editing. With respect to our collaborations and the research and development that we do, we emphasize that the original idea of the service came a bit out of despair, because we were running out of money and funding opportunities. So that was a major driving force. But in parallel, we thought that it could be a good idea to use these projects also to develop our own tools and make papers out of these, because we have been working on the technique itself, and we have been publishing on that. So if we make a project for the client and at the same time we can use that project to validate, to make a proof of principle using some new type of technology, that could be a win-win situation. So with respect to collaborations, we are quite happy to have started a few. And I'll be telling you about 4, just in the interest of time. The first is actually the first one that we thought of. I don't know if Katerina is there listening to this presentation, but several months ago we discussed this idea of improving on a methodology which relies on the fusion of Cas9 with a monomeric streptavidin, and as you'll see in a minute, this has a number of advantages in terms of increasing the efficiency of the editing, and there is room for improvement here, and we are trying to improve it, as you will see, and to combine also this type of technology with a new trick that we call dream catcher CRISPR. It's very experimental. It's basically still an idea, but we'd like to test it. Then there is a project involving the immune pluripotent cells and splice mutant variants with phenotypes in cardiomyocytes. This is one of the main projects of the group of Carmo Fonseca, the IMM, and they recently got a La Caixa grant, in part based on results that we produced. So we are very happy with that, and we'll be continuing our interaction. What we do mostly are knock-ins, either to introduce a mutation or to correct a mutation, and this is mostly the work of João Proença, senior postdoc in my lab. He has been working very hard on this for the past year. And in the future, we expect also to start using more cutting-edge technology, not to introduce mutations, but to play with the transcriptional activation of the genes without changing the sequence. Then there is a project in which we plan to master this orange method, that is a type of CRISPR protocol in which the homology recombination that I showed you before in the context of the original work by Capecchi is not used. What's used to insert the DNA is a non-homologous joining, and this will be more easy to understand in a minute also. And then we are trying to establish a CRISPR editing system using this C3P3, which doesn't tell you much, but it's basically an engineered polymerase, RNA polymerase, that boosts the transcription levels of whatever gene you want to express in the cells. And we thought that it could be perhaps interesting to do it for CRISPR. So now a bit more in detail with respect to the first project. The idea is twofold. We want to improve on this method in which streptavidin, monomeric version of streptavidin, is fused to Cas9, and because of that we can link this template DNA, which is biotinylated, and it's going to be bound to the streptavidin, and this way you can increase the local concentration of the template, and as a consequence you increase the frequency of targeted events. It works very well in mice. We're not sure if it's going to work as well in cell lines, but we'll give it a try. And what we want to do now is to improve this method by introducing 2 point mutations that have been described to increase the affinity of streptavidin to biotin, and perhaps this will have consequences for the efficiency of the technique. So that's project number 1. Project number 2 is a bit more ambitious and risky. the idea is to do a variation on this technique by using as the DNA piece that is bound to the streptavidin, not the template, but essentially a platform made of a single stranded DNA, onto which we will have several copies of the same template single-stranded DNA being ligated just because of the homology between the sequences. And this way, at least in principle, we should be able to increase the local concentration of the template. Because as you can see, here the template and the Cas9 are in a 1-to-1 relation, whereas here it could be 1-to-6, 1-to-7, and we'll see if the system works. But that's basically the idea that I would like to try. We call it a dream catcher because this structure, at least to me, it reminds me of the dream catchers of North America. And I would just like to make this point, although we have been talking about this idea, namely the one of the 2-point mutants, for a while. We finally started doing it. So we have a master student, Maria Gonçalves is developing this project. She's cloning these two mutations. They are actually in this PCR band, and we are cloning it as we speak. And then the idea is to do a first set of assays using plasmids that encode the Cas9 fused to the streptavidin, the monomeric form, with or without the point mutants. And if we get encouraging results, then we'd like to purify the protein and perhaps start using it in the different platforms that Congento has to see if it works in zebrafish, for instance. Then we have this project in which, with Carmo, besides the bread and butter, which is the knock-ins that we have been doing, we'll also start to play with the transcription levels of the genes without editing them. This is by no means a new technology. It has been around for a number of years, and it's mostly based on fusion proteins, again, in which Cas9 is fused to a transcriptional activator, and it's actually a dead mutant version of Cas9 that will not cut the DNA, but will guide the transcriptional activator near the promoter so that transcription can be improved. Recently, there was a paper describing a variation on this method that we'd like to implement, in which Cas9 is not fused to a partner. Instead, it's the guide, which is modified and has this extra loop that will attract this transcriptional activator. And it seems that this type of variation on the original technique has a number of advantages, and we'll be trying that in the context of our collaboration with Karma. Then there is this project which we are starting with Edgar Wunsch, also from the IMM, in which the idea is to master this orange method, which was also published recently in the context of making knock-ins in primary cells, which is one of the requirements of this project with Edgar. And what's cool about this technology, as you can see, is that it's really very elegant because you use the Cas9 guide complex in two ways: to make the break, as it normally happens in any CRISPR-Cas9 project, but then you also use it to release the donor template from its plasmid by cutting it on each of the sides. That's releasing this tag. And then the insertion is going to be made not by homological recombination, so you don't have these big homology arms, which are sometimes difficult to make or can be expensive if you order them. No, it's much simpler. You just release the tag, and then you expect this type of repair mechanism that the cell has. So non-homologous engineering basically recognizes double-strand breaks, such as this one, and it will ligate the breaks so that in the end you can have this type of structure and the insertion of your tag exactly where you wanted it to be. So we are excited to start this project. I'm not sure if the efficiency is going to be limiting, but we'll see. And finally, we will also establish the Cas9 system using this C3P3 platform, which basically uses an engineered RNA polymerase to boost the transcription levels and also the processing of the RNAs. And this is just a comparison of what they can see using luciferase as a readout. You can see that the levels of transcription that the system achieves are much higher than what you get on a normal transfection. And that's essentially what I had to tell you with respect to our concerns and future goals. From our experience of the last year, we came to the conclusion that although the service has been moderately successful, and we got several clients and we are making some money, and it seems to be a sustainable service. In fact, if you factor in salaries, it's not yet there, and it's very difficult to create a service without some support from the institution. I'm talking about CEDOC, FCM, also Congento that gave us some help, or any other institution. I think this is critical to, at least in the first years, to gain momentum and then start becoming more independent. And because FCM charges 25% of our revenue in overheads, we don't really have a big margin to grow as a team or to buy equipment that would allow us to go to that next level. So we are quite limited at this point, and this is very, very frustrating. However, I think the service has already shown that it's a viable option. Clearly, we expect to improve in terms of efficiency, and that these efficiency gains will also free some money that will help us grow. Our immediate goals are to ensure that the projects, these 30-something projects, are all completed. So far, I have to tell you, we only completed 2, but we have all the other projects running, and we expect to complete most of them within the next 4 months or so, and to get a good client retention. If that happens, we may secure a number of clients that will sustain the service. At the same time, I've just been informed by the Broad Institute, that has one of the patents of the CRISPR technology, that we can actually have as clients biotechs, including big companies and startups. And this was a restriction before. We thought that it was not possible, but in fact it's possible. We just have to pay some royalties to the Broad Institute, but the percentage that they charge is not that high, or in any case our price is so low that even raising the price to take into account the royalties will get us a price that is still very competitive. So this week we are starting a campaign of massive emailing, which is our way of doing campaigns, to try to get clients from companies, and I'm expecting to broaden our number of clients by starting to target also biotechs instead of only people from the academia. And finally, we will also start applying for external funding to try to solve the problem of the salaries, which is, I would say, our Achilles heel for now. And this will happen probably in collaboration with similar CRISPR services in Europe, with which I've been in contact over the last year. And that's it. If you have any questions, great. If you would like to order us a line, even better. So let's hear from you. Thank you.
Thank you so much, Vasco, for your presentation. We'll now stop recording and we